final talk this morning is Robin Polt from University of Arizona. You have
I think I'm all set.
Good.
Can you guys hear me? I'm not sure this stage is rated for my weight, so I'm going to walk down here if you don't mind. Oh, the clicker. Where's the clicker? Ah, so I do have to go up there.
Here you go.
Thanks. I appreciate your indulgence here. At any rate, I've been trying to tell this story here for about 25 years now, and we started out saying, well, we can make opioids, and I think we're now to the stage where we can say this is a general process, and I want to try and convince you of that. Oh, why did you do that? Yeah. At any rate, we would like to have everything be orthogonal, and you can say, oh, well, maybe this is organization and this is chaos, or vice versa. But there's a tendency, I think, you want to have one drug that hits one receptor that has one effect, and for one disease. And in fact, life's not like that. And although it's useful, I think, to tear things apart, we have to remember to put them back together. And I guess that's one of the points I want to drive home here is the non-orthogonality of these things. Lots of people have been looking at the PK/PD of peptides and peptide drugs for a while. It's good to see pegylation is of interest. Glycosylation is not on this list. And I hope to change that. But one of the things, of course, GLP-1 analogs are very useful. It's been spread out to a lot of different companies now. But, in fact, you remember where this comes from? It comes from a Gila monster, saliva of a Gila monster. And so, in a sense, nature has done a lot of the design work for us. And so we're starting sort of on third base, when we make these home runs. There's an error here. This should be 4,000 molecular weight. The thing I don't really understand, and I'm just because I'm just a chemist, I guess, is this volume of distribution, and what does it mean when a creature, which is presumably a human, has a 55 liter volume of distribution. I mean, okay, so we can call them apparent volumes of distribution, and they probably have some use, but I'm not sure that they're really useful in the concept, in the context of peptides, or it's easy to overgeneralize, I guess is the point I'm trying to make. So our starting point are the neuropeptides. Typically, the process that we're advocating is you identify the peptide sequence and its activity. Generally speaking, this has been done for us. I mean, there's more than 250 sequences identified. Unfortunately, a lot of the activity is defined from outside the brain, but that's okay. It's still a good place to start. Now, the other key thing I want to point out here, and I think it's critically important, is these are not orthogonal processes. If you say, okay, I'm going to optimize binding, and I'm going to get the best binding drug in the world, and then after I've got that, I'm going to go back and modify that to make it transport well, that's just a fool's errand. You really need to do these simultaneously and be considering PK/PD from the very beginning. Here's a few targets that I've identified or pulled out. I don't know that they're, CRF and GLP-1, GLP-2, a lot of these are known, opiates, of course. Anyway, I'm going to try and focus on 4 little stories here, and I apologize in advance that these are great stories, but they're mysteries in some cases. We got started some time ago making enkephalin analogs, and we got this paper published after a couple of tries. It's pretty reasonably highly cited, generally because people say, oh, GLUT1 transporter is involved, and let me point out it is not involved. We never showed any evidence that it was. That was just the impetus for us to make the compound in the first place. It's basically the physical chemistry that's directing this. And so in that paper, we point out that morphine 6-gluconoride is a good prodrug, and it's basically amphipathic. Its inactive isomer here is also inactive. But, and of course, if you put your sugar in the wrong place, you block binding, so you have to be sensible about this. You can't just stick the carbohydrate anywhere you want to. And generally speaking, water solubility is all you need to achieve here. We've made a number of glycopeptides that don't have a sugar on them. So you can put a phosphonate or a sulfate on there, and you get water solubility. And you can see that's still being explored. This is a very recent paper in which they just put a sulfate here. Interestingly, this converts it to a delta agonist instead of a mu agonist. But I still think there's still quite a bit of controversy about this compound and how it gets into the brain. Here's a review where they actually do include glycosylation. And we have enhanced peripheral effects. I guess that's a limitation. But in fact, if it's cleared from serum, glycosylation can become an advantage if you consider the long-term effects of the drug versus the short-term effects. Let me drill down on this glycosylation. I can summarize this here. Glycosylation gives these things drug-like behavior. They basically become active. Having the endogenous ligand as a starting point really gives you a big advance. I think I've already made that point. And finally, the point that I think really isn't going to be appreciated until you get into the clinic is the fact that these drugs are degraded to amino acids and sugars, and you don't get a cascade of metabolites that are different in each person depending on their compound. I think we really have to get into the clinic to see that. Another point I want to really drive home here is we're not penetrating cells. We're not going into the cytosol. Topologically, we're always outside the cell. Of course, you get an endocytotic event, you're physically inside the cell, but topologically, you're still outside. So if you have an exocytotic event, you've now passed through the cell without ever going into the cytosol. The cytosol is death for us. As I said, people are catching on to this. There's nothing special about O-linked glycosides. You can have N-linked, and you can have C-linked. And Toth has written a nice review that's just now coming out. I think it's worth reading. So here's our 4 stories and a vignette. Let's start with glycosyl Damgo. Of course, Damgo is well known as a mu agonist. Here's a NMR-derived structure in the presence of a micelle. So in the presence of SDS micelles, we find peptides adopt very restrictive conformations. And here you can see the glycinol part down here. And basically, we're just going to modify this part of the molecule to achieve our goals. So we can construct a Connolly surface by asking Mo to do that in Mo, the software, and we can get a surface area of the lipophilic portion, surface area of the hydrophilic portion, and then if we put this in the form of a equilibrium constant, we call this our amphipathicity value. We can calculate that. It's pretty straightforward. So if we take and replace that glycinol with a serine amide, and then a serine amide with a 5-carbon sugar, and then a serine amide with a 6-carbon sugar, and a serine amide with a disaccharide, you can see we grow that hydrophilic part, and the membrane-bound portion remains the same. And we can actually quantify that. We can use DOSY NMR and actually look at these SDS micelles. The ones that are loaded with glycopeptide move slower than the ones that are naked. And we can actually get these binding coefficients down here. And it's really hard to measure these things in the 10 to the minus 2 region. This is one way you can do that. And we can plot that versus our amphipathicity value, and we get a reasonably straight line here. You can also just use reverse phase HPLC retention times. That gives a good prediction, I guess, of lipophilicity. I'll say more about that. In the mouse, if we administer these to mice, and I apologize, the order here is reversed. So DAMGO is this point, and our lactocele DAMGO, if you will, is this point over here. And so what you see is as we increase the hydrophilicity of this compound, we go from inactive compound to an active compound, and then we get less activity. Okay, and this is just a tail flick data here from a mouse versus our amphipathicity calculated values, and that's all published here, so quite a while ago, 10 years ago almost. If you don't buy this amphipathicity stuff, you can use a hydrodynamic volume, which the carbohydrate chemists have been using for decades. But you still get this U-shaped or V-shaped curve that's easily understood in, why did they do that? Easily understood in the sense that if we have a very small hydrophilic portion here, we spend most of our time on the membrane. Well, that's great if you're a neurotransmitter and you only have to go 100 angstroms from presynaptic membrane to a postsynaptic membrane. But for drug delivery, that's terrible. So you're spending all your time in the membrane and you're not going anywhere. On the other hand, if you have too much carbohydrate, you spend a lot of your time in the aqueous compartment and you don't hit the membrane, which you need to hit in order to A, bind to the receptor, and B, undergo endocytosis so that you can cross cellular barriers. What you want is to optimize that and get somewhere here in the middle where you can hop along the membrane. And so what I'm advocating here is that our drugs, after they're administered, rapidly distribute to membranes, and then they hop from membrane to membrane to membrane, hopefully eventually finding the receptor that you want to find. And so optimization to achieve membrane hopping is what we want here. Let's look at the dose response curves. Here's LIM 100, here's morphine. I don't think just looking at these curves a pharmacologist could distinguish these two compounds. Look at the doses here though. This is about 40 times more potent than morphine on a molar basis. And basically, my student who made this, Larissa, warned us, these compounds are toxic because I got stung by one. And so if you've been working with peptides, you typically don't worry about them just going up your nose, but these will if you're not careful. At any rate, so this is basically like fentanyl with a long-acting duration of action. Gosh darn it. I blew it, didn't I? I'm sorry. Can you still hear me? Yeah, so we don't want to make a compound that's 20 times more addicting than morphine. It's, that compound has real high self-abuse potential. So we switched to this compound, and which we're using a Roche sequence here to make a delta selective drug. So that's D-alanine is now D-threonine, no N-methyl group, and we've got a leucine here. And this is the lactoside. So we call this lactomorphin. And to make a long story short, the Navy paid us some money to develop a less sedating analgesic for use on the battlefield. And I think we've got it. It's right here. And we made two batches of this, or we paid to have two batches. One was made by Lonza. The other batch was made by Polypeptide Labs. And so I think at one point we have about half a kilo of GMP grade material in the minus 80 freezer, and we still have a good deal of that. So if you can think of a great use for lactomorphin, I could be happy to send you a 5-gram jar. Here's the tail flick data, 55 degrees C tail flick in a mouse, and you can see there's morphine, small dose, medium dose, large dose. There's our drug, small dose, medium dose, large dose. very potent analgesic in the tail flick. There's the area under the curve. And this, again, this is milligrams per kilogram here after sub-Q administration. So we're crossing several barriers there from subcutaneous space into the bloodstream, then the bloodstream into the brain. Beyond simple tail flick, you can look at more robust models of pain. The writhing, here's a mouse writhing. And the way this is done is the student administers the drug, videotapes this, and then another student who's blinded to this looks and counts the writhing. You can see little marks on the tail there so they can keep track of which mouse is which. And you can see this, you inject a bit of acetic acid, induces the writhing, and then doses of the drug will reduce that writhing, and it looks very much like morphine, essentially identical to morphine. So we have, I think, a very potent drug here, lactomorphin. It is, of course, orally active if we use the secret sauce. And I don't think there's anything special about the ones we have here except that we're honor bound not to tell you who gave us this or what it is. But that's pretty decent oral activity. So people were concerned that lactomorphin had too much kappa activity. So, okay, let's make one that's pure delta. So we went outside the human brain and we used a dermorphin-derived drug. And you'll notice there's not 4 amino acids here. It's tyrosine, D-alanine. That is a naturally occurring D-alanine in the salamanders and frogs that produce it. Then a phenylalanine. So essentially the phenylalanine, the tyrosine just can't reach far enough to activate the kappa receptor. There it is. It's highly delta selective, 14 nanomolar at delta. It's micromolar at binding at mu, and there's our kappa binding. Nothing, nada. And of course it is an agonist. So we thought this would be a useful drug for chronic pain. We still do, or inflammatory pain. You might ask, and actually we have a company that you can talk to about this. But you might say, well, what the heck is the carbohydrate doing? This stuff is already pretty active. Well, here's the compound with no sugar. Pretty gelled up there. And so you don't have to be a pharmacologist to see, oh yes, our carbohydrates making these compounds more water soluble. Lipophilicity is an issue, and I would regard BBI 1108 as a highly lipophilic peptide. Here's some examples. I've actually worked with this compound. It's more soluble in chloroform than water, which is kind of unusual for peptide maybe. Here's a nafarlin, which is also in the same context. And I guess this is maybe just a pure personal thing with Lipinski. I was giving a talk one time, and it had this great audience, and I was about to tell them all about my glycopeptide drugs, and everybody got up and left, and there's like 12 people left. I said, what's going on? They said, oh, Lipinski's talking in the room down the hall. And so, at any rate, don't be blinded by this guy. We can make these things pretty easily. Here's some HPLC traces, and so you can vary the lipophilicity to your heart's content, I guess. So here's BBI 1108. You see 14 nanomolar. Just dink around with this a little bit, from glucoside to celebioside, mannoside, and you see we can get even better activity here. Compare that to morphine in the complete Freud adjuvant mouse. So this is a inflammatory pain model. I guess hopefully you're familiar with it. You inject CFA into a paw of a mouse. It swells up. It gets inflamed, and then you can do a hot plate test, and you can use the uninflamed paw as a control. And of course, morphine's pretty effective, and so is our drug. You can do tactile allodynia in that inflamed paw, and there's morphine. Here's our drug. And you can go to rats, and so you do a sciatic nerve ligation to to create chronic pain. And of course, gabapentin is effective there, huge doses. And with the BBI compound, same sort of thing, thermal hypoalgesia. Anyway, so it looks pretty good. We're concerned about side effects, GI transit of morphine. You know, of course, it blocks down. This is a charcoal meal study, so you give the mouse his last meal in charcoal. You wait a period of time after you've given the drug, and then take out the intestine. You just pull it out and measure how far that charcoal has traveled down the gut. And you can see morphine stops that flow. Our drug has some modest effect. And look at these doses. These are huge doses, and the mouse tolerates it. You can also do a colonic bead expulsion experiment, which I'm told is the first experiment all pharmacologists do, at least in Bilski's lab. And here's compared to the lactomorphin. So this is here. And you see lactomorphin, more like morphine than it is like the pure delta compound. And it shows all the appropriate inhibition from naltrexone and so on. But look at the serum concentrations. You administer this drug. I think this is IV. Yeah. And you see, boom, it's in the serum. Boom, it's out of the serum. And so we're seeing effects, though, out here. And so clearly it's got nonlinear PK/PD. And I used to think that was somebody else's problem, but it's my problem now. It is, without the secret sauce, you see it's modestly orally available. With the secret sauce, it's reasonably available. So we think this should be studied further in a clinical context, I guess. Finally, you know, when we got money from NIH to study our lactomorphin, we said, well, you know, we can also do endorphins and dynorphins. And we didn't really think this was true, but we thought, you know, it makes a nice little golly gee thing to put at the end of your proposal. So we started looking at this, and of course your molecular weight's getting up there now at this point. And anyway, we were, to spare you a whole lot of stuff, we did a lot of experiments looking at the conformations of lactomorphin in the presence of my cells, really quite rigid. Similar sorts of things with the endorphins. We started out calling them endorphins. It's not clear that they're endorphins. They're probably more akin to dynorphins. And in fact, well, I'll say more about that in a minute. But anyway, there's a lot of conformational interest here. And so I think the point I want to make here is that if you want to know what the conformation of your peptide drug is, you may well want to consider doing it with deuterated SDS, not an aqueous solution, because the aqueous conformation means nothing. We weren't the first people to notice this. E.T. Kaiser published his paper back in '84 when he noticed the amphipathic helix of the endorphin molecule. And we've sort of mimicked that. And if we do the same sort of treatment with Mo with the surface, you can see there's a lipophilic surface and a hydrophilic surface, and it's quite helical. And the idea is if we have peptide versus monosaccharide versus disaccharide, we adjust the hopping rate here along the membrane. The other way we can do this is just look at the intrinsic stability of this helix. So if the helix is real stable, it'll tend to be bound to the membrane. If the helix is not real stable, it'll pull off the membrane. We published a paper on this. I don't think anyone's read this yet. They certainly haven't cited it, but it's worth reading anyway, I think. We took our helix here, and here's our same lactomorphin, Roche sequence, tyrosine, D-threonine, glycine, phenylalanine, leucine. And here's our address sequence, and all we're doing here is we're adding 4 methyl groups to these guys, well away from the message segment. So we have gly-gly, AIB, AIB, alanine, alanine, and then everything in between. And if we look at the helicity of these compounds by circular dichroism in water, they're all more or less random coil. In the presence of trifluoroethanol, You see the helix forming there, a trend. And if you go to 50 millimolar SDS in water, buffered water, of course, you see even more trendy trend. And here, so that's data the data you just saw is this red bar graph here. And you can see we can go from very helical to not so helical. And if we look at the disaccharide, it shifts it. And if we go to no saccharide at all, so this is the native peptide, you see the same sort of trend. This guy here is in purple because we can't measure its NMR in water because it's not water soluble. It just forms a helix and falls out of solution. But we can measure it in the presence of SDS micelles. It goes right into solution. Companion paper there, we took this a little further and looked at this this linker, for example. And what we find is if the linker is flexible, then the amphipathic helix can satisfy the membrane binding requirements. The message can satisfy the receptor binding requirements, and everybody's happy. If the linker is stiff, then you get less binding. And then if we mess around with this helix and make it maybe more than just an amphipathic helix, maybe it participates in binding. It's cut through a lot of stuff. So here's our Roke's message. Now we've got a flexible GABA linker, GABA aminobutyric acid, and our same helix that we've seen before. If we do an ICV injection where we're bypassing the blood-brain barrier, you see that's pretty potent, 46 picomoles per mouse to get an 850 value. If we do IV injection, you see there's small, medium, large dose, and we're down here at 280 picomoles per mouse. That's IV. That's per kilo. And if we take this data, compare it to the unglycosylated case, there's 10 milligrams per kilogram of the unglycosylated material. Just not going in anywhere. Look at the same data, compare it to the compounds we've already seen, morphine, our lactomorphin, and now our helix-modified lactomorphin, and you see we've gone from 6 to 4 to 0.28 micromoles per kilo in the A50 value. So unfortunately, we couldn't suss out the difference caused by stability issues and transport issues, so we actually had to start measuring transport values, and Bickel was nice enough to publish this paper. And you can sample whole tissues, you can do microdialysis, you can do, I guess, put catheters in the subarachnoid space. I don't know exactly how to do that. All of these things, though, typically require determining volumes of distribution. But we've decided microdialysis is the way to go, and we think it works pretty well. Typically, you insert a dialysis tube, and then you allow this to heal overnight or 24 hours, and it works just fine. So Omar Mabrook, a postdoc in Bob Kennedy's lab in Michigan, did this for us. And basically, here's our injection, IP injection. I don't know why. But anyway, you can see that it enters the bloodstream, and then goes into the brain, and you can see we get a spike, and then it levels off to a reasonably high value of drug. That's like 500 there, right? And if you look at endogenous peptides at the same time, you can see there's leuenkephalin, same time, so there's the injection. And you see, it's not at 500, it's down below 50. So we're getting drug in the brain. Again, to cut a lot of stuff out of here, we've gone all the way from things at 500 molecular weight up to over 3,000. Generally speaking, there's no correlation between molecular weight and transport. It's entirely up to the amphipathicity of the drug, we believe. Angiotensin, a nice little compound. If you're interested in the pharmacology of this, contact this lady, Meredith Hay. Don't talk to me about it. But basically, there are more than one angiotensin receptors, And angiotensin 1 through 7 is of interest. It's a great model peptide for us because it breaks in half, fragments really nicely in the mass spec. So very sensitive. So here's a glycosylated version. My colleague Mike Hine and his student Kat Kramer did this. And so here we just put a carbohydrate on the end of this angiotensin 1 through 6, I guess. So it fragments into mass spec here. Carbohydrate's first thing to come off, and it breaks in half, and it breaks differently in half, I guess. And we get ions like this. So this is these collision-induced dissociation, and so you can see we get pretty good fragments there. So without much collision, you see mostly parent ion, and as you have more collision energy, you start to see more of the fragments. Oh, I thought it was me for a second. You can get these up to 100% and then fragment these further. And so in this case, we only need to use ms to the second. For other cases where they don't fragment so nicely, we have to use ms to the third to actually identify them. But using this technique, you can get serum levels and CSF concentration levels from the same mouse at the same time. And this is really nice because you think about it, injection site variance is normalized between animals. Makes a lot more sense than trying to do the typical BUI sort of analysis. This is small dose. This is after 20 minutes, so it's just a single point. But serum concentration, CSF concentration. So this is after we've already hit peak values. And this is log scale. If we take that off log scale, you can see it looks a little more impressive this way. Oh, there's still a lot of variation here. This is an N of 5 mice, I think. But clearly, we're getting the drug in the brain, and basically you have 20 minutes of high concentration in the serum, and during that time, your peptide goes wherever it wants to go. Stability is an issue, of course, and the native angiotensin 14 minutes, and these are serum, mouse serum taken out of the mouse. So this is just in a vial, right? 14 minutes, we make the amide, it's more stable. Glucoside, even more stable. And then these disaccharides, we get very stable. And so we thought 60 minutes would be okay for this initially, but we ended up having to run it for 8 hours to get stability from that. We're not the only ones to see this. Istvan van Toth, who was spending a lot of time putting lipophilic moieties on his peptides, is now a true believer in glycosylation. So this luteinizing hormone, releasing hormone, can be stabilized by changing residues, of course, and you get full from 4 minutes to 19. But you put the lactoside on there, you hit the big time there in terms of stability, and it's orally active. So I think we aren't the only ones now, and that's good. Real quickly here, just describe this collaborative arrangement we have to cure Parkinson's disease. Not just treat it, but cure it is the idea here. But we have Hine doing our stability and actually measuring things. We make these things. We have a guy, John Stryker, who started out collaborating with us from UNE, but actually we brought him into U of A. And then we have a Parkinson's expert doing this as well. But very quickly here, stability, not a big issue. This is mass spec data, and you can see the points where these things cleave. The interesting thing about this is this is coming out of the CSF of a mouse, not via the normal way. And then these glycosylated compounds have activities. Here's an example. And so these are pretty good sized compounds at this point, but they seem to work. Now these are class B GPCRs. They bind not only to the transmembrane part of the receptor, but there's this whole 250 amino acid N-terminus of the receptor that binds the helix part of these things, and then this jams the message segment down into the transmembrane part. And of course, all that's missing in the opioid, so you just have the transmembrane part. And so this makes it very easy. Let's say if you just truncate this, now you got something that binds to this part, but not to this part, and that's not sufficient for activation. So you can make agonists by this way, and until they bind to both parts of the molecule, you don't get activation. And so if you have it truncated, you should be able to block endogenous drug from, or endogenous PACAP from activating. And anyway, I can't say much more about this. The glycosides bind, you can see. Of course, if these are EC50, so presumably binding is buried in there somewhere, but the efficacy is good enough that it must be binding pretty well. And you can see if you take these PC12 cells, let them grow, they just divide and form clusters. If they're grown in the presence of PACAP, instead of growing, they start developing. And of course, our glycosylated compound has the same effects. And the idea here is, that we can affect, I think, microglia activation in the brain and hopefully treat a number of things. And so I want to thank you guys, and I thank all of these guys who did all this work, and all of these guys who did a lot of other work, and these guys who paid for some of the bills.